Good morning, one and all. I am Dr. S. Manigandan, Assistant Professor of Botany, Srividya Mandir Arts and Science College, Kateri, Uttangarai. Today I talk about sexual reproduction in angiospermic plants. Sexual reproduction. It is a three parts: pre-fertilization, fertilization, and post-fertilization. What is the reproductive organ in plants? Flowers. Reproductive organs and the structure of a plant. Most flowers are hermaphrodites. It contains a male and a female sex organs. The flowers, what is it? Thus, a flowers is a specialized shoot that is determinate wise indeterminate. As a modified stem with compressed internode passes modified leaves with various functions, this determined by gene arrays. Example ABC model, often clustered in an inflorescence, large branch. Parts of flowers: corolla, calyx, stamen, pistil, receptacle, and peduncle. Stamen male sex organ and pistil is a female sex organ. Reproductive parts for reproduction in main parts of a flower, stamen and pistil. Stamen, the male reproductive organ, collectively called androecium, and composed of two parts, anther and filament. Anther, expanded lobular structure at the end of the filament. It function produce pollen. It is a male sex cell. Anther and structure. It is a bilobed dithecous longitudinal growth. It is a four-sided tetragonal for microsporangia. It is called pollen sac. Anther wall layers: epidermis, endothelium, middle layer, and tapetum, and final one is sporogenous cells. Anther wall layers is first one is epidermis, one cell thick. It function is resistance of anther. Stomium, present between two pollen sacs. Anther dehiscence at this stomium region. Endothelium, below the epidermis, one celled thick fibrous cellulose thickening plus a little pectin. It functions at maturity. These cells lose the water and help in a dehiscence of anther. Tapetum, innermost layer surrounds the sporogenous tissues. Cells are multinucleate and polyploid, contains ubious bodies. Ubious body secretes sporopollinae. It functions as a nourishes to the developing pollen grains. Microsporogenesis. It is formation and differentiation of microspores is called microsporogenesis. This is a sporogenous tissues into a sporogenous cells. It is a diploid cell. Then it produce microspore tetrad. It is haploid. And microspore tetrad, pollen grain tetrad. It is also haploid. Then final microspores, pollen grain. Structure of pollen or microspore. It over 60% of angiosperm pollen grains are shed at this two-celled stage. So, what is a part of pollen? It is one is vegetative cells, then another one is generative cells, and inner layer is intine, outer layer is rough and exine. Then gem 4 is present in the pollen grain. Sporopollenine. It is a resistant to high temperature, strong acid and alkali enzymes, and it is a fossil because of the presence of sporopollenine. The female reproductive organ and the unit is called pistil. Pistil or carpel. Both are called gynoecium. The female reproductive organ, collectively called gynoecium, composed of three parts: stigma, style, and ovary. Stigma, swollen tip of that pistil. It functions covered by sticky substances for pollen grains to adhere. Stick, specialized for receiving pollen grains. Style, it functions long slender tube that connect the stigma to the ovary. Act as a conveyor carried from one place to another of pollen grains. Ovary. The ovary core is solid structure at the center. Placenta. The edge of the core nourishes the egg. Structure of ovule. It is a sporo. It is a megasporangium. Cross section of an ovule. The ovule has the following parts: integument, nucellus, funiculus, embryo sac, micropyle, and final one is chalaza. The integument is a two-layer protective coat. The nucellus is a mass of tissue consisting of meristematic dividing cells. Funiculus attached to ovules to placenta. Embryo sac, it is contain the egg. Types of ovules. It is 6 types. It's have. So one is orthotropous, anatropous, hemianatropous, campylotropous, amphitrapous, and the final is cysteinotropous. Orthotropous ovule is straight, and this is the most primitive type of ovules. Example plants piper and polygonum. Then anatropous ovules, it is turned at 180 degree angle. This is the most angiospermic plants is found in 90% of families is it have. And final one, third one is hemianatropous ovules. Ovules turned at 90 degree angle upon the funiculus or body of ovule, and it has it and is at right angle to the funiculus. Example ranunculus. Campylotropous ovule. This ovule is covered more or less at the right angle to funicle. Micropyle end is bent down slightly. Example in members of Leguminaceae and Cruciferaceae. Amphitrapous ovules. The ovules as well as the embryo sac is covered like a horseshoe. Example Lemna pupae alisma. And the final one is the cysteinotropous ovules. The ovules turns at more than 360 degree angle. So funicle become coiled around the ovule. Example Opuntia, Cactaceae family, and another one is Plumbaginaceae family. Development of gametophytes. Egg. It is a egg. Two stages. One is megasporogenesis and another one is megagametogenesis. Megasporogenesis is a beginning and creation of megaspores, and megagametogenesis is a creation of gametes. So for example egg. Development of gametophytes. Egg. Megasporogenesis. It is a ovule produce megaspores mother cell. It is diploid condition. Then megaspore mother cell produce 4 megaspores via 2-time meiosis. It is haploid condition. The 4 megaspore is produce megaspore mother cell, but 3 megaspores die and only one is left to continue. And another one is megagametogenesis. The surviving megaspore divide via mitosis 3 times, forming 8 haploid cells. Egg development in ovules. Among the 8 cells, 3 cells migrate near the micropyle. The one at the center become the egg, while the other 2 become the synergid cells. The other 3 migrate and the opposite pole and become antipodal cells. The remaining 2 cells are naked, no membrane, and remain at the center to become the polar nuclei. Development of monosporic embryo sac. Monosporic embryo sac is called only one megaspore is functional group. It is called a monosporic embryo sac. Megasporogenesis is 3 types. One is monosporic, then bisporic and tetrasporic. Monosporic is Functional megaspore form the female gametophyte or embryo sac. This type of development is called a monosporic development. Example, polygonum type. Then bisporic. Bisporic of four megaspore formed if two are involved in embryo sac formation. The development is called bisporic. Example, allium. And the final and the third one is tetrasporic. If all the four megaspores are involved in embryo sac formation, the development is called tetrasporic. Example, peperomia. This is the formation of embryo sac. One is megaspores and one is monosporic, and another one is bisporic, and last one is tetrasporic. The tetrasporic example plant, fritillaria. Egg development in ovule. These 8 cells form the inside of the embryo sac and are ready to be fertilized by a pollen from the male. Thank you.